Hi, thank you for your presence today. Thank you for the committee to invite me today. This is part of my thesis. I just finished my PhD last month, so let's see. Yeah, thank you, I have this. So first, this is the first paleoproteomics work developed, research here, made in Israel. So let's see how this works. So first, what is paleoproteomics? Paleo is Asian. Proteomics is the branch of biology that studies the set of proteins that are expressed by a genome of an organism. Okay? So during archaeological excavation, we find a lot of animal remains, what we call zooarchaeology remain. So archaeology is the branch of archaeology that studies the relationship between humans and animals. One important change in this relationship was the domestication of animal, okay? The process of animal domestication. That started in the north, the Fertile Crescent, what is today Turkey, north of Syria, Iran, Iraq, and was adopted as a new idea by the southern Levant, okay, that we are here. And we are talking about the period of the pre-pottery Neolithic. This is before ceramics, okay, just after the domestication of plants. So all this work that I show you are based in three main markers. First, we see an increase in the increased frequency of animal remains, including of non-local animals. In our case, it's going to be the Bos primigenus, that is the ancestor of the mother cattle. Then the second marker is going to be change in morphology. For example, in our case, it's going to be the decrease in size from Bos primigenus to modern Bos taurus, what we call today, what we know it. And the third change is the change in the natural mortality profile. This means that we are expecting a natural mortality profile to have 50% of each of the sex, okay? Without sex bias. However, in a modern Earth management, I'm going to expect a change, okay? For example, in the case I will expecting to have much more females adults, okay? And one or two, a few males, because I need less males for reproduction. But we can learn more about it. We can understand also the motivation, why this process started, as to obtain more milk, to obtain more meat. That's the big question, okay? Because for both of them, I'm going to expect very different mortality pattern. For example, for milk, as I said before, I'm going to expect in the adult remains to have a majority of females and a very small number of males. However, if I want meat, I'm going to expect to have all the animals in the size of the most bigger size, for its much meat, and without the sex bias. I don't care if it's a female or a male. I want the meat. But what are the tools that we have today in zooarchaeology to study sex? First, we have the osteological analysis that we are going to compare between different bones of foreign collections, okay? But we have here the problem of sexual dimorphism, so we need to be, we need better tools. Of course, we have Asian DNAs, and this is the most informative of all. However, in our area, Asian DNA is really bad preserved. This is a very arid climate, so Asian DNA is really hard to extract. We have paleoproteomics, and this is something new from the last 15 years. The most used technology methodology is the ZOOM, so archaeology by mass spectrometry, but this don't give me the sex, only the species. So what are the alternatives? What we can do to know the sex? We are going to apply paleoproteomics on enamelon or tooth enamel proteome. So this is the tooth. The enamel is the most mineralized organ in the body. During the development of the enamel, I'm going to, during the development of the enamel, there is several proteins that are produced for the control of the mineralization. And we are going to have a small, 1% of proteinose materials on the enamel. During the ameloblast are going to produce different proteins, but we are going to work only in one, in amelogenin. Why? Because this is a dimorphic protein, have a copy in the chromosome X and the chromosome Y. And during the evolution, there have been several changes, mutation in the sequence between the X and the Y chromosome. So if I can pick, I can extract and found a unique peptide that is only in the males, I will know the sex, right? All the work I will show you in the next slide is based in the work that was done already in 2017 and 16 by Stewart, and he found the way to do it in humans, extracting. This is the amelogenin Y, amelogenin X. You can see that they have very conservative sequence, but there is some mutation between them. He found that in position 59, the methionine in the amel Y is oxidized and is missing in the amel X. So he developed this methodology for humans. So all this question I saw you, all the motivation, we have to know the sex. And this is why we developed a new methodology using label frequentification to determine sex in bovines. And this was already published in last August. In this paper, I presented the sample of 8 mother samples and 12 samples from the Isamun from the pre-pottery Neolithic. Okay? Now I will show you also 15 samples from Abu Ghosh, from a very similar period, from the middle pre-pottery Neolithic. How I did this? First, in our laboratory at Venancio building, plant science, okay? But we are from archaeology. We extracted, cleaned this. I cut a clean piece of enamel. I cleaned it from the dentin for everything calculus, and I put it in HCl. We have a demineralization, and we send the sample to our collaborators in the proteomic unit. They will be peptide desalting, centrifugation, then it's run for the nano liquid chromatography MS, and after that I will receive thousands of peptides that are present from the amelogenin. I found unique peptides that are only presented in the males. So what I found, 2 unique peptides that are for amel X, 2 unique peptides for amel Y. In the left we have the sequence. And how I presented this result. First, in the amel Y axis, you have the intensity of one of the amel Y unique peptides. In the x-axis, I'm going to have the intensity of one of the amel X peptides, amel Y and amel X. So all the samples I have in the x-axis are females because I don't have an intensity for amel Y. And then the samples that are males, I'm going to found it with intensity in amel Y and amel X. Okay? Here are the mother samples, and these are the archaeological samples. I did this for all the peptides, one X again one Y. And let's talk about the important things, the sex ratio, what I found in the archaeological samples. So first, for the archaeological sample of Abu Ghosh, on the middle pre-pottery Neolithic B, we see 60% female, 40% males. Okay? This is something that, this is not milk, we can say, right? This is not one and all the majority females. And what happened after, in the next period, in Beisamun, in the transition between the pre-pottery Neolithic B and C, again, still this trend for more females are getting, higher. However, when I go to the pre-pottery Neolithic C from Beisamun also, this shift change. Now I see more males than females. Okay? So what we can say about the motivation for domestication? Let's see. First, in summary, we developed a new methodology for sex determination in bovines. And I don't have time here, but we did the same for sheep and goats. But what are the motivations for domestication? So to obtain more milk, to obtain more meat. We can understand that, I didn't say this, but the samples that we have are adults in the maximum size. So we can say that was to obtain more meat and no more milk. And I would like to acknowledge and to thank you to my professor, Elisabetta Boareto, for giving me the opportunity to develop this incredible methodology, and all the zooarchaeologists and archaeologists that give me the samples, and of course our lab group. So thank you.